there is a thread-like differences between transgenesis and cloning. What is mean by transgenic, sir? Genetically modified organisms. Transgenesis is a technique by which genetically modified organisms are produced. Genetically modified organisms are produced by a technique is called transgenesis. Okay. Genetically identical organisms are produced, that is called cloning. Genetically identical organisms. Technique by which genetically identical organisms are produced is called cloning. Technique by which genetically modified organisms are produced is called transgenesis. Is it clear? So transgenesis is the process of introduction of extra DNA, that is foreign DNA or exogenous DNA, whatever it is, extra DNA is introduced into the genome of an animal to create and maintain the stable heritable characters in a newly formed organism is called transgenesis. So here genome of an organism is altered by introducing a small piece of gene or DNA, so that it is called as genetically modified organisms. Genetically modified. My cell, it is my cell. It is my own cell. Not cell phone, it is my own cell. It's a body cell. It contains diploid set of chromosome. 46 chromosomes is there. Okay. If you add one more chromosome into the genome of my cell, now the cell is called a genetically modified cell, because the original genome of an organism is changed. Either benefit or harmful, that is differ. But here altered, so that it is called transgenesis. The foreign DNA which is introduced into the cell or introduced into the tissue or introduced into the embryo or into an organism, such a foreign DNA is called a transgene. A gene which is inserted into a cell, that is called a foreign gene. Animal, from the cell animal is produced. Such animal is called a transgenic animal, or genetically engineered organism, or genetically modified organism. Transgene, by which an organism is produced. Such organism is called a transgenic animals. You see, steps involved in production of transgenic organisms. First step, identify and isolate the desired gene. Desired gene. The gene function is known. Known function gene is isolated. You take this, the known function gene. Oh, can you give me the example for known function gene? As in this slide it is given, the gene which is responsible for secretion of more milk in organism. Such a gene is isolated. This is the desired gene. Desired means I do like the gene. Gene is more because it induces the organism to provide more milk to us. So desired gene is isolated is the first step. Then suitable vector is isolated. This is called a suitable vector. Nowadays isolated. Then combination of desired step is known to contain my desired gene. So that it is called a rDNA, recombinant DNA, or recombinant plasmid. Recombinant plasmid is the right answer. Recombinant plasmid, sometimes we do say it is a recombinant DNA also, because the desired gene is introduced to bind with the DNA of the plasmid. So rDNA or recombinant plasmid. Now it is can be accessed into or introduced into either cell or tissue or embryo or adult organism, any organism. Now it could be introduced. But in this case, I do introduce into an embryo or ovum. Is it clear? So ovum is isolated from the goat ovum, in which the desired gene is injected. Now it is called as genetically modified embryo. The genetically modified zygote is injected into the surrogate mother, uterus of the surrogate mother. After the gestation period, the goat gives a new young one. In this new young one, my desired DNA is also known to found. Such a new gene starts to produce its particular protein by which it induces the organism to give more milk to us. So milk from transgenic code is more and more, that is quality, quantity, sweet, everything will be there because such a gene is identified, analyzed, and I fixed this is the most best gene. If it is injected into any organism by which a new organism produce, which will give the best milk to us, I do already know that. I have assured that, so that I isolated such a desired gene, injected and produced a genetically modified organism from which what I need fulfilled. Is it clear? This is steps. First step, what is the first step? Isolation of the gene. Second, vector isolation. Third, combination. Fourth, injection into this organism. Either directly it could be injected or indirectly. Vector, if you use the vector to inject the desirable gene into gun is available. You keep the desirable gene in a particle gun and shoot directly. The gene is injected into the embryo or adult organism or tissue or cell. That is called direct method. Okay. So nowadays, mice, rat, rabbit, pig, goat, cow, sheep, fish, these are all the organisms have been produced in a transgenesis method. Any doubts? This is steps. Definition I have given you. Then I explain what are the steps involving in a production of reverse transcript, transgenic organism, and then uses of transgenic organism, uses of transgenesis. Transgenesis is a powerful tool. Is a powerful tool for what? To study the gene expression and developmental process in higher organism. By the transgenic animals, we come to know the gene expression. Either it is good gene to induce the organism to give more milk or not. Suppose the gene may be responsible to synthesize the insulin, by which I may come to the gene is suitable gene to induce an organism to produce the insulin or not. So gene expression will be identified with the help of this technique. Then transgenesis helps in improvement of genetic characters in animal. Genetic character of the animal will be improved with the help of transgenesis because the desired genetically qualified gene or DNA isolated and cloned in a laboratory and then used to increase the genetic character of the organism by inserting into it. And also it helps us to understanding the human diseases, by which it helps us to invent the new treatment for the new diseases. So any medicine is discovered directly, it could not be injected into the human for the testing. So transgenic animals are produced, that is used as a human being, in which we can inject our new drug and by which we can they will respond. By their response, we come to know whether it is a good medicine to control the corona or not. First step, it will be given to the animal, then it is practiced in the human being. You know already this is. So it helps us to understand the human disease and produce the novel approach in the treatment. As well as it also exhibits some human diseases, anemia. These are all the diseases related to the human being. So some genetically modified organisms also can exhibit such a diseases. So by giving the treatment, how are their response, everything could be understood with the help of the transgenic organisms. Then transgenic animals are used to produce the particular protein which are important for medical and pharmaceutical application. Here the protein refers to insulin, human growth hormone, factor 8, whatever it is. 13 factors are associated with the blood clotting. All the 13 will be produced in a laboratory with the help of transgenic animals. So protein will be produced. Some drugs are produced, antibiotic, monoclonal antibody, whatever it is used. Organisms used as a host to production of this protein and used to rectify the diseases in human beings also. Whereas transgenic mice are used to testing the safety of vaccine. Yes, if you find any new vaccine against any diseases, use the mice as a host to test the safety of vaccine. Then testing of toxicity of the drugs. Any vaccine, any drug, anything you found a new one before giving to the human being, use the transgenic animals as a host in which it should be properly tested, then only it is transferred to the human being. At the same time, the transgenic animals are more sensitive to the toxic substances than the non-transgenic animals, so that we use this as a best host for testing of the drugs and vaccine. So transgenesis is important for improving the quality and quantity of the milk, meat, egg, wool production in addition to testing the drug resistance. So these are all the uses of transgenic animals. Anything else? Transgenesis, I have given a definition, then steps, then uses of transgenesis. Move on to the biological products and their uses. Product which is produced from or isolated from any living organism, such a product is called biological product. So we are going to see some other biological product with their uses. Animals or transgenic animals are used as a bioreactor. Bioreactor is nothing but factory. Okay. Organism uses a factory to synthesize more number of desirable protein. If vaccine for the COVID-19 is discovered, okay, COVID-19. So such a vaccine could be produced in tons, then only it is practiced or it is used to provide to the human being. Such a quantity of the vaccine is needed now. Here the organisms are, transgenic organisms are used as a factory to produce the desirable protein in tons. That is, for example, antibody is a biological product. Antibody is the, here microorganism, macroorganism, whatever it is used as a bioreactor. Okay. Antibodies are the substances, you know that already. Antibodies are the substances or metabolic product of one organism that react against the disease causing pathogen. So antibiotic is a therapeutic agent. It's a metabolic product of one microorganism used to inhibit the metabolic product of other microorganisms. So it's a uses of antibodies. Then monoclonal antibody is a biological product used to treat the cancer and heart related diseases, as well as solve the transplantation problem, rejection problem. Transplant rejection problem is also solved by monoclonal antibody. Monoclonal antibody is the antibody one who used to treat only the particular disease. Used to treat against only particular disease is called monoclonal antibody. Monoclonal antibody is available to treat the cancer nowadays. Growth hormone induce the growth of the standard children. Then rDNA vaccine induce the adaptive immunity. You see in the red color, whatever given in the red color, they are considered as a biological product. The black color is their uses. You see here natural protein adhesive. Adhesive refers to attach. A protein used to attach something with something. That is called protein, adhesive protein. Okay. They are non-toxic, easily biodegradable. Non-toxic, easily biodegradable. Rarely trigger an immune response. Hence could be used to reattach the muscles with the bones by using tendons. Reattach the tendons and tissue. Sometimes filling the cavity in the teeth and repair the broken bones. For example, I would like to give the natural protein adhesive. Filling the cavity in the teeth. No, have you heard that in a root canal treatment, adhesive protein is there. That is Cutapersa resin. Cutapersa resin mixed with zinc and bismuth oxide. Such a combination used to seal the cavity of the tooth. In a what is called root canal treatment. That's adhesive protein. Tendon used to attach the muscles with the bone. Ligament is the connective tissue, connects the two bones. Okay. Already used. Know that. Human alpha lactalbumin. Human alpha lactalbumin is a recombinant protein used to produce more milk, used to produce milk more similar to breast milk. You think that it is used to, is a recombinant protein, it is used to produce the more milk which is similar to breast milk and promotes the gut health. Gut means digestive system health. Also it is used to induce the